Good morning, and welcome to the latest edition of our ATS Southern California exclusive Zoom series. This morning, we are honored to feature superstar Technion professor Shulamit Levenberg, whose work has left its mark in several different disciplines, from tissue engineering to regenerative medicine to diabetes and even 3D bioprinted meat, and whose work has been creating a lot of buzz in the media. For those who don't know me, I'm Raj Tasman, board member of the American Technion Society in Southern California. Allow me briefly to share with you my Technion story. My husband, Herb, who passed away in June, was an estate planner in the Philadelphia area and involved with the American Technion Society. When we moved to Laguna Niguel in 1998, I became involved locally and learned even more about the Technion. I feel strong ties to Israel and believe in its well-being. The Technion has contributed more than any other Israeli university to the security, infrastructure, and continued success of the country. When I learned of the amazing accomplishments of the Technion that they had achieved, it resonated deeply in me. And as a humanist, I was impressed that Technion shares its knowledge and innovations with others around the world. In addition, its unique integration of science, technology, and medicine has helped the world immeasurably. And this is why I am so passionate about the Technion and a proud member of the Technion, the American Technion family. In addition to our community in Southern California, I am delighted to welcome friends from Seattle to San Francisco, to Park City, to Potomac, and beyond. We may live in different areas, but we share a passion for the Technion. Together, we are investing in scientists and entrepreneurs like Shulamit, whose research and innovations impact Israel and the world, our health, and even the food we eat. I would like to extend a warm welcome to Mark Chavan, and I also would like to give a special welcome to Rina and Avner Schneur of Boston, who have generously supported the Rina and Avner Schneur Center for Diabetes Research, which Professor Levenberg heads. We are so happy that you can be with us today. I'm also pleased to announce that Schneur family and Professor Levenberg lab are putting up $1 million, provided that we can raise $500,000 in additional donor support. This will go towards further advancing Professor Levenberg's promising research in diabetes. If you're interested in learning more about this wonderful opportunity, please let Pamela or your local director know, and we will be glad to share additional details. It is now my pleasure to introduce Professor Shulamit Levenberg. She is the former Dean of the Faculty of Biomedical Engineering, Technion's largest faculty and the largest and oldest of the state of Israel. She holds the Stanley and Sylvia Sheridan's Chair in Cancer and Life Sciences, and also has the Technion Center for 3D Bioprinting, which she launched in March 2019. She is the recipient of numerous awards, including the Crow Prize for Excellence in Scientific Research, awarded by the Wolf Foundation. She founded 2 startup companies, including Aleph Farms, is a member of the Israeli National Council of Bioethics, and is actively involved in training young scientists. Professor Levenberg earned her bachelor's degree at the Hebrew University of Jerusalem, her PhD at the Weizmann Institute of Science, and conducted postdoctoral research at MIT before joining the Technion in 2009. A few housekeeping rules before we begin. Please make sure that you mute yourself during the presentation. We also suggest you watch this in speaker view mode. After the presentation, we will have time for questions and answers. At that point, we ask you to to literally raise your hand and we will call upon you. Or you can type in a chat question in the chat. When we call on you, please then unmute yourself. It is now my honor to turn this program over to Professor Levenberg.
Thank you very much for the nice introduction, and thank you very much for every one of you for joining us today. And I would like to share some of our research, some of our studies. And for this, if it's okay, I will share my screen. 1 second. Can you see it okay now? Okay. Okay, so we'll start. My main focus in the lab is tissue engineering, and tissue engineering evolved as a way to find solution to shortage in organ and tissue donation. When some tissue or organ are damaged, you have some alternative, but all of them are not so good. One is to get a donation from another person, but then the patient needs to get immunosuppression through all the life. And we also know that there is a problem in getting organ and tissue donations. And another solution is to use the patient's own tissue, but then you have to sacrifice one area to regenerate the other part. So you can take a bone from one leg to regenerate bone in another leg, or take a vein for a bypass operation, or piece of a muscle from one part to repair another part. But these operations are, of course, very complicated. And actually we have now 2 injury sites instead of 1. So tissue engineering tried to find a solution by engineering the tissue from the patient's own cells, so we can isolate the cell, the stem cells, and then we can culture them in vitro and expand them, and then grow them on 3D scaffold. You can imagine a sponge or a gel where the cells are growing in and proliferating and then maturing into the tissue. And then we can take this tissue and implant it to repair or replace the damaged tissue, and the scaffold itself can be biodegradable and can disappear over time, and you can left with a native regular tissue. So this is very, very exciting. And for this, usually when we know that tissue is made from more than one cell type. So in the lab, we decided to take several cell types together to engineer the tissue. For example, if we're talking about skeletal muscle tissue, so we can take the muscle cells, we can take endothelial cells that form the blood vessels, we can take support cells, and we can mix them in the vial, in the tube, after we find the right cell concentration and the medium composition, and then we can seed them onto the scaffold. They will get into the pores of the scaffold, and then they will start to organize themselves, to proliferate, to organize, and to form the tissue structures. And one of the problems in tissue engineering is how you really feed the implant after you implant it. And you need blood and to get into the graft. So we and others develop a technique where we can add endothelial cells, and the cells, we find that they will form spontaneously blood vessels that will form a network. And you can see it in the animation, the formation of the tube. And if we add support cells, they will be recruited to the tube and differentiate to smooth muscle-like cells and stabilize the vessels. And if we add myoblasts, stem cell of the muscles, they can form the myotube, they can form the fiber. And by this, we can get vascularized muscle tissue that we can then use to repair a damaged muscle. So if we want now to use this tissue for implantation, what can we do? We can take the construct, and it can be vascularized with the whole network of vessels. And we can now use it to replace damaged muscle. For example, here, it's abdominal wall piece of muscle. And we can suture the construct, the engineered muscle. And then we were able to show that the engineered vessel really recruited vessels to get into the graft and facilitate the blood perfusion into the implant. And because we see also connection between the engineered vessels and the host vessels, eventually these engineered vessels can become functional and blood can flow relatively fast to the inner part of the graft, of the construct. So we can get nice regeneration and nice perfusion, and the muscle can integrate very nicely into the surrounding muscle and repair the problematic area. So this is how it looked under the microscope. You can see here the graft area, and the engineered vessels are labeled in green, and everything here can be filled with other cells, for example, the muscle cells. And what you see in blue are the vessels, the host vessels, that are now getting inside slowly into the implant. And if we have the engineered vessels, it will be much faster. And in red, we can inject fluorescent dye. Here we injected a dye, a fluorescent dye in red. And this will indicate all the perfused vessels. So if we see red dye in the vessel, it means that blood is flowing through them and they become functional. So if we now look at the magnification, we can see, we can see the very nice connection between the host vessels and the engineered vessels. The engineered vessels are in green and the host vessels are in blue. And you can see that they're really very nicely connected. And if we now go and want to see if they're functional, we can look at the red dye and see that, yes, indeed, some of the engineered vessels, the green one, have red dye inside them, meaning that they really became functional and blood is now flowing through them and they're helping to perfuse the implant. And really seeing this was really very, very exciting for us to see. And this is just an example of another study. This was already accepted. I have to change it here, but for publication. And what we were able to show here, that we can vascularize bone implant to repair bone damage. So what you see here in red are all the vessels that are going now inside into the bone area that is in white and feeding the bone, so it can really help to regenerate area where there is a real defect that cannot repair by itself, and we need to implant something inside the bone to allow its full recovery. So to move to larger pieces of tissue that can be clinically relevant, we added the bioprinted printing, and we actually established the Bioprinting Center at the Technion. And what you can see here is an example of such a bioprinter where the ink is not a regular ink, it's a bio ink. We can print the scaffold, and we can also print the cells. So we have different heads and we can print different cells in different locations. What you see here is also an example of forming a nice vessel, a tube inside that will become the blood vessel, the main blood vessel of the tissue. And we can again feed the cells as you saw in the animation. We can add endothelial cells that can form the network. We can add muscle cells, and we can add support cells. And eventually we can build the tissue now that can be exactly in the shape of the injury and fit exactly to the injury site. And this is another printer that we have in the lab, demonstrating some more features. Here we print into a bath, and we also see the more complicated structure of the vascular network. This allows us to define the exact structure that we want, and also the exact location of each cell type in the tissue. So bioprinting is really becoming part of tissue engineering, and we are happy to be a part of it. Of course, it involved, the center involved, major printer and manpower to help other researchers also to come and print what they need. And the center is open to all researchers at Technion and the country and industry, and people who want to bioprint can come and we can help them do it. So how this engineered tissue, or later on bioprinted tissue, how they can help in patient, how they can really become relevant in the clinic in the future. So a very important example is in diabetes treatment. And we were able to show, using the vascularization technique, that we can vascularize pancreatic islets, and these can be implanted and better survive in diabetic mice. And what you see here is showing here the islets, the cell that secrete insulin can be implanted, and then they can secrete insulin and respond to glucose. And when they are vascularized, they can survive. And if we implant them under the skin of diabetic mice, they can really reduce the blood glucose levels. Instead of a type 1 diabetes patient that have to inject insulin all the time, the cells can stay there and survive and secrete insulin in response to glucose. But recently, we also showed that we can use tissue engineering to treat type 2 diabetes. And this was a project that was really, we were able to do it by the generous donation of Avner and Rina Schneur, that they donated the money to perform this project, which was really out of the box thinking and very risky, but thanks to the donation, we could really go ahead and do this project, which eventually came out to be a very, very successful project. And we are very happy about it because it really goes to patients. And I just want to briefly describe the project. What we do, we can now, because we know how to engineer muscle, we can take muscle cells, but we can also overexpress the glucose transporter. Because we know that in type 2 diabetes patient, the insulin, the response to insulin is not so good, and there is insulin insensitivity, so glucose is not getting into the cells. So what we did, we added the transporter and created a skeletal muscle tissue, engineered muscle that can now absorb glucose better from the blood. We implanted it into the muscle, similar to what you saw in the animation. And what we saw that first we have to show that the cells really express the transporter, and you can see it here. And in vitro, this piece of tissue can absorb glucose in better way. And you can see here in the graph, they can really have glucose absorption and reduce the glucose level in the surrounding medium. But then we took and implanted this piece in the abdominal wall muscle, and we looked at the tissue, and I hope you can see here very nicely that the blood glucose level really went down just by implanting this piece of engineered muscle, because it changed not only this small piece absorbed the glucose and removed it from the blood, but it had a systemic effect on the whole metabolism that really cured the mice from the diabetic situation. So you can see here the piece here implanted. This is the construct in vitro, and this is the implanted piece. And the systemic effect can be shown here. You can see these are staining of the liver, and all the red dots here are oil droplets. And what you see here, this is the control, and the one here in the right are the mice, a sample of the liver of the mice that were implanted with the engineered construct. And I hope you can see that you have less oil, less fat in the liver. The liver is not so fatty. And you can see it also here in the graph that it's really affected the whole metabolism. It's really affected the situation of the animals and cured them from the diabetic situation. And here you can see example of another model, another diabetic model, where the mice are, they have a problem again in the insulin sensitivity. And also here we can see when we had the glucose tolerance test, we can see that the mice that were implanted with the construct didn't reach so high glucose levels. They really were, the glucose level were stabilized and also went down very nicely. So these results are really, really very exciting, and we really hope to move now to a more human model and to move this project closer into the clinics. And we are now hoping to get more donations to be able to do this project, to move to human system, to show that we can have an implantation procedure that can fit human, and hopefully it can help to many, many diabetic patients. Another aspect of our work is a spinal cord regeneration. We wanted to see if we can engineer a construct that we can implant and it can repair the spinal cord. And the hope, of course, is that the paralyzed patient can start to walk again. So the model that we used is a rat model, and you can see the paralysis in the lower part, the lower legs, the back legs of the rats. And you can see that they are really paralyzed, and they will not have any spontaneous repair, similar to what we see, unfortunately, to what we see with patients that are paralyzed. And what we did, we took the construct, the engineered construct, and seeded it with stem cells that can provide neurotrophic factor that can help regeneration to occur. And you can see here, this is after a few weeks, 2-3 weeks, you can see that the rats really started to walk again. And this happened only in the treated group. And again, this was really, really very exciting. We did not expect such full recovery to almost normal situation, normal motor activity. And this also led us to another project where we want to do it more clinically relevant. And we, beside adding blood vessels, as you saw in the animation, that can really have more vascularization and better recovery, we also moved to a more clinically relevant model where instead of really implanting a construct in the spinal cord, and this is very invasive and the surgery is a little bit, complicated. So we moved to, instead of, implanting the cells, we decided to give the rats exosomes, which are vesicles secreted from the cells, and we used modified exosomes that were given to the rats through the nose and reach the spinal cord area. And also here, what you see here, the green line here showing that they had a much better recovery and started to walk again. Just by this intranasal administration of this exosome, the cell particles that are loaded with siRNA that is now helping to have more axonal growth and more regeneration in the spinal cord. So this is also very exciting, and we also want to move this to the clinic and try to help paralyzed patients. So we see that tissue engineering is really can give hope to many areas in regenerative medicine. And we actually founded 3 companies out of technologies that were developed in the lab. One is Nurexon that I mentioned for spinal cord recovery, and the company is now raising funds. And we also founded NanoSynex. NanoSynex is about microfluidic device for antibiotic susceptibility test. And I didn't talk about the technology that we developed that was the base for this company, but the company is also very, it's a startup, but very successful, and with the hope to bring down the time that one have to wait till the doctor decide which antibiotics need to be given for the patient. And another company is Aleph Farms, which I will mention now. And Aleph Farm is actually based on the technologies that I described, creating skeletal muscle engineered tissue, but instead of using it for medical application, using it for food application. So the concept is very similar, but now we just get cells from the cow and use edible scaffolds to grow the cells on. And then the cell will differentiate to muscle fiber, and eventually will get in the plate a piece of skeletal muscle tissue, and it can also be combined with fat, and we can also actually get a piece of meat that we can engineer. And based off this technology, where you can see here very nicely the muscle fiber that are formed, and these are already fiber from bovine fiber from the cow, we made a whole project. This was a project that was the idea of one of my students that is a vegan, and he really wanted and came and asked if we can use the technology that we develop in the lab for tissue engineering, for muscle tissue engineering, if we can use it for meat. And his PhD was on combining the different cell types together, and we were able to show also here that if we have more than cell type, not only the muscle cells, but if we add support cells and we add the endothelial cells, then the tissue will really resemble better the natural tissue, and we'll get better differentiation of the muscle. And Aleph Farm was founded, and you can see here a piece of the minute steak that was developed in Aleph Farm in the company, and the chef, cooked it, and people already tasted it and said that it's really nice and very, very similar to the regular steak. These are thin pieces of meat. This will be the first product of the company. And then we moved to the next step using 3D printing. And what you can see here, this is the printed steak, the bioprinted steak, where we're able to develop a bio ink that we can use to print the muscle cells and fat cells, and with a printer, we can, as I said, we can decide where the cell will be and how they will be localized with different heads of the printer. And what you see here, the different area, the red that resemble the meat and the white that are the fat area. And this is after cooking. And so again, this is very exciting and showing that we can use tissue engineering not only for medical application, but also for other application. Of course, for me, it's not the main direction. I'm still focused on the medical application, but it's nice that something else can come out of it, and the company can now continue and work on the production of the meat. And we're just developing more and more techniques and developing the technology and understanding how the tissues can form for the various aspects. So I would like to end here and thank, of course, all my students that took part and in the different projects. And I want to mention Rita Backerman, who worked mainly on the engineered muscle for type 2 diabetes, and Shawei Ju, who worked on the spinal cord regeneration. And I mentioned Tom for the muscle, and also Yedidia and Iris and all the other students. And of course, all our collaborators. And I want to thank all the funds that allow us to do the project. And especially here, I want to thank Rina and Avner, Schneur for their fund that allow us really to do this really, really exciting project. And also the Shervington Fund for allowing us to do the spinal cord project. So thank you very much. And if there are questions, I'm happy to answer them now.
All right. Okay. Shulamit, thank you so much. There really isn't anything you can't do. We are incredibly fortunate to have you with us today and to have you part of the Technion family. Before we open this up to Q&A, I have a few questions of my own. Alternative meat is quickly becoming a growing business. What drew you to this field, and what other areas of the food industry do you see expanding to?
What was the first part? I couldn't hear you.
Okay.
Well,
what drew you to the field of alternative meat, and what areas of the food industry do you see this expanding to?
You know, I'm not expert in the food industry, so it will be a little bit difficult to say, you know, broadly to answer this question. But I think that really we will see more and more of tissue engineering getting into this, and also bioprinting, food bioprinting will get into it. So I think we will see a change or additional of other aspects of the food industry, something that we're not familiar with them now, but I think we will get more and more familiar with them.
Why do you feel that the Technion is a leader in 3D printing?
Why do I think so?
That the Technion is a leader in 3D printing? You know,
3D printing is something that is evolving now around the world. So we're not the only one who are doing 3D printing. We also opened a center for bioprinting, but for sure there are other centers around the world, but each center contributes more projects and new ideas and more techniques. And we are also contributing some to this really important area.
And being the superstar that you are, I'm sure that you had many options of universities to establish your lab. And we're really very lucky to have you at the Technion. Why did you choose the Technion, and what makes Technion unique, especially for women, to study science and technology?
Okay. So I can say that I really chose Technion because I wanted this interdisciplinary direction, and I wanted the combination of engineering and medicine and biology. And the Technion, the Faculty of Biomedical Engineering, is really the largest in Israel and the most established. And so this was the most attractive place for me to come and continue my work that I started at MIT in tissue engineering, to continue it in the biomedical engineering faculty at the Technion. So this is really, I think, the best place in Israel to do this type of research. And overall, the students are really, really very, very good. So it's a real pleasure to work with the Technion students, even comparing to students that I saw in many, many other good universities around the world. So it's a real pleasure to be at the Technion.
Well, your enthusiasm and your skills are really wonderful, and we appreciate having you. And I'm going to open up Q&A to everybody. Please raise your hands and we will call on you. Please be sure to unmute yourself.
I think Howard and Barbie should have a question.
Yes, I do. That was a fabulous presentation. It's amazing that you're doing work in both diabetes type 1 and type 2. It's just incredible. I'm wondering if you have a feeling for how many years away we would be from actually printing solid organs such as kidneys or livers.
I think that functional organs really are more of a challenge than pieces of tissue. But in this organ, people are talking now on printing the functional units of the organ, similar to what we do actually with pancreas. We work with the functional cells that can do the important function of the organ. So I think that we will see it also with liver and with kidney. And I think we are getting there. There is still some work to do, but I think in the future we will see also more complex organs that are being printed. The printing is really giving us more tools to position cells. And when we have in these organs, the position of the cells and the complexity of the different cells is really very critical for the function of the organ. So with this tool, I think we are closer to reaching the goal, but of course there is a lot to do till we reach it.
And when do you think you might transition from developing tissues from mice to humans?
For muscle?
For muscle.
So this is exactly what we are trying now to do with what we call the Schneur project, the type 2, the engineered muscle project, where we want to move now from engineering the mice muscle to using the same technique but to work on human tissue. So we're doing it now. We're starting to work on it now, and it's something that require additional work to prove it, and we're on our way.
Do you need any volunteers?
First, we need the funds to do the research, then we will need the volunteer when we reach clinical trials.
Thank you. Thank you so much.
Any other? I'm looking for hands. Mark, stay fair.
Thank you for taking my question. I'm interested in how you would expect to deal with the autoimmune component of type 1 diabetes. If the disease is the result of the attack on the beta cells in the first place, how would you deal with controlling that, suppressing it, whatever the words are, when you replace the beta cells or the islets, even though they're made from the patient's own cells? That's what caused the autoimmune reaction in the first place. How do you see that playing out?
So one direction is that some labs are working on is isolating the islets. So you implant them, but isolate them so the immune cells cannot reach them. So you put them in a type of a polymer or material that can isolate them. from the attack of the immunocells, because you're right, this is one, the problem that we see in type 1. Another direction is because we know that actually the right connection, the tight connection with the blood and the signaling that the cells need to get from the surrounding tissue, because this is very important. Another direction is actually not so much to isolate them, but to integrate them well with the tissue. But this may require, implanting again and again, maybe once a year or once in 2 years. But if you have a stock of cells like pluripotent stem cells, induced pluripotent stem cells that can be isolated from the patient, but then reprogrammed to the pluripotent state where you can really multiply the cells and get more and more and more of the cells without the need to really get more cells from the patient. You just have a stock that you can grow and differentiate. Then it's not really a problem to do the repeatable implantation after 1 year or 2 years, but then because it takes time till the body will attack the cells and that they will stop really functioning. So these are the two modalities that people are aiming to in implantation of islet.
Thank you. Thank you.
Great. I think Richard has a question.
First of all, fantastic presentation. Thank you very, very much. I wanted to ask you if there are any applications for cartilage regeneration in the areas of arthritis.
So yes, tissue engineering also deal with cartilage. So there are projects on cartilage. We specifically started to work on cartilage in the aspect of ear reconstruction. And we have a project together with a physician from Sheba Hospital where we 3D bioprint ears using cartilage that we can get from the patient. But the same concept of cartilage repair and cartilage regeneration can be then used for other problem of cartilage.
Okay, thank you.
Great. And I think we also have some questions in the chat. So one of them was asking about how long do innovations like the ones that you're working on take to get to market in other countries? And when do you think Americans will see the benefits of your research in their local communities?
I think usually actually America can be sometime before Israel because Israel is waiting for the FDA approval and then approving in Israel. So it can be in parallel, so no worries. It will reach United States, I think similar to the time that it will reach Israel. But again, how long it takes, depending on the project, depending on the funding, depending if a company is being established, then it can move much faster. So then, it depends.
Thank you. And then there are other, I'm looking around for hands and hand raising. I don't want to miss anyone. Oh, I see another question from Mark.
Yes, this question relates to the laboratory-produced meat concept, the olive farms programs. To many people, a laboratory raised meat is desirable because it avoids harming animals. To others, it's desirable because it might lower the carbon footprint. Yet, for most cell cultures, you use animal products, you use fetal calf serum, you use casein hydrolysates, to use all kinds of things that do involve animals. And you also use carbon dioxide to regulate the pH and all kinds of things, sterility and air processing that take a lot of energy. Is it really serving the two primary goals, animal well-being and low carbon footprint, or is there some other motivating factor in this kind of program?
Of course, there are some other motivation factors. It can be the food can be without antibiotics and can be with the right fat, combination. It can be healthier and everything. But yes, of course, the animal well-being and the world well-being is really two main things that this area is aiming to, and it can avoid animal suffering and benefit our, the climate changes and all the ecosystem. And what you mentioned, the need to use animal serum, for example, you don't need to use serum, and now people are using serum replacement. And also Alefarm is not using any animal component in the culture of the meat besides the cells, and that can also be harvested without harming the animal. And also, when you mentioned about the carbon footprint, it was shown in many studies now that it's really much less than growing the cows and all the what's part of this process. So even the fact that you need to use CO2 for the incubators it's totally less. It's really less than the farming.
And the bottom line, of course, is, can it be cost effective?
Yes, so there are now many studies showing that yes, it can be cost effective, and Alefarms already say it will be very similar, maybe a little bit more expensive, but similar to the prices that we're used to now.
Yes, and kosher.
Yes, kosher. Why not? Of course.
Thank you.
Any other? I'm doing one last scan of hands. Okay. Well, if there are no other questions, I just wanted to thank Shulamit again so much for taking time out of your very, very busy schedule to be with us today. It is always so great to learn and hear about the work that you're doing that really is impacting lives around the world. You, as you said, you really truly exemplify the multidisciplinary work of the Technion and really are an inspiration to women scientists and new generations of female scientists. So we thank you, and we look forward to hearing updates about your work and your successes, and just also know that you always have an invitation to visit us here in Southern California. So we hope that we can see you in person and not just virtual. For those who don't know me, I'm Pamela Wohl. I'm the ATS Senior Director of Development in Southern California. And I want to again thank all of you for being with us today. And I hope today leaves you with a renewed pride in your support of the Technion. If you have any questions, including the one about the match, please feel free to reach out to me or your local ATS staff person. And we look forward to seeing you at future SoCal programs. In the meantime, our next national webinar is on May 20th at 9 a.m. Pacific, 12 Eastern Time with Technion alumnus Oded Gal, who's the Zoom Chief Product Officer. So you can register for that at ats.org/events. And thank you again for joining, and we hope that you enjoy the rest of your day. Bye.
Thank you very much. And you're welcome to come and visit the lab when everything will be back to normal.
We look forward. Thank you.